So now the floor is for Rodolfo Goya from Argentina. Hello, Rodolfo.
Hello.
Yes, normally, yeah, sharing the screen will be no problem, I think. Go ahead.
Okay. Okay. Well, first of all, I want to thank the Hela Foundation for inviting me to participate in this meeting. Today, I will speak about mostly about rejuvenation by cell reprogramming, and to a lesser extent, I will speak about two experiments of life extension in our laboratory. So the story of biological rejuvenation began over 5 decades ago with the discovery of animal cloning by John Gurdon and collaborators in England. Then there were other important discoveries in the meantime, but the other seminal discovery happened a few decades after Gurdon's studies, and was the discovery of self-reprogramming by Yamanaka and Takahashi in Japan. What these Japanese researchers demonstrated is that the transfer of 4 master genes, KLF4, SOC2, OCT4, and CMYC, now known as the Yamanaka genes, the transfer of these 4 genes to a culture of skin fibroblasts from mice after the 15 days of action, more or less, of the genes, turn these cells into cells that behave as embryonic stem cells. So they called these cells induced pluripotent stem cells, and this started a revolution in biology. And so it also paved the way for the implementation of self-rejuvenation by cell reprogramming in cells from all humans and all animals. These are very nice experiments. There are a number of them, but we don't have the time for me to describe any of them. So the researchers next moved to the in vivo scenario. Let me see if I can move my screen. Okay. So the first approach to rejuvenate mice in vivo was published in 2016 by the group led by Juan Carlos Belmonte at the Sol Institute. They used progenic mice, which are mice very short-lived because they age prematurely. They live less than 1 year. So, these mice were also transgenic for the 4 Yamanaka genes under the control of a regulatory promoter. So they used old animals, which were less than a year old, but were really biologically very old. So when they added the antibiotic doxycycline to the drinking water of the animals, they turned on the 4 Yamanaka genes, and cell reprogramming began in the whole animal. But 2 days later, they removed the antibiotic from the water, and they interrupted that reprogramming process and silenced the 4 Yamanaka genes. They let the animals rest for 5 days and then added doxycycline again. Reprogramming began in the whole animal, but 2 days later, they again removed the antibiotic and so on. So this is called cyclic partial reprogramming. And after a number of cycles of partial reprogramming, these researchers studying visceral, mostly stomach, spleen, kidney, and skin, observed that many of the aging signs, histological aging signs in these organs and tissues were attenuated or right away eliminated, disappeared. So they also observed that the treated animals had survival curves 50% longer than the untreated controls. So this was definitely a seminal experiment, a proof of concept that rejuvenation could be achieved in vivo, although at that point, very limited because it was just the beginning, but was a quite important experiment. And very recently, an experiment was published by Jesus Avila and Manuel Serrano, Spain, working in Spain, using the same kind of model that Belmonte used, except that they used normal mice, not progeria animals. So they, in the rest was the model was very similar. So they performed in these mice cyclic partial reprogramming for several months, much longer than Belmonte treatment, until the animals reached middle age. And they were interested in memory and hippocampal function and structure. So they studied that in the animals, and they got really modest results. So I would say almost disappointing because I really had a lot of expectation about such kind of experiment. So this is proof that trying to implement in vivo rejuvenation using transgenic animals for the Yamanaka gene probably will be a quite challenging endeavor. And this paper by Serrano and Avila was published last year. And also last year, another paper, very interesting paper, was published by the group led by David Sinclair at Harvard University. It was published in Nature. So what they did was gene therapy for 3 of the 4 Yamanaka genes. They used OSK, they left out thymic, constructed an adeno-associated vector, type 2, regulatory vector. So they injected the vector in the eye of normal mice and waited for some time, and then pinched, crushed the optic nerve of the animals. This affected vision dramatically, was really quite deteriorated, the vision of the animals. So they added doxycycline to the drinking water at this point. This action turned the 3 Yamanaka genes on for several weeks. So, and then near the end of the experiment, they performed an optometric test on control animals and untreated animals that had been exposed to the Yamanaka genes for several weeks. And they found out that the control animals had still a very poor vision because of the damage of the vision, but the treated animals have recovered vision quite significantly, really quite significantly. And so they also studied, after sacrificing the animals, the optic nerve of the controls and experimental animals. The asterisks here are indicate the place of the crush of the axon. This is the optic nerve. And they found that in the controls, there was no axon regeneration, but in the animals that were exposed for several weeks to the OSK genes, there was a clear axon regeneration. And if they continue to exposing the cells, the retinal cells, to these genes, the regeneration continued really and was quite extensive. So this was really was a gene therapy approach. It did not intend to rejuvenate the whole animals, but the important significance of this achievement is that they showed that they could express the Yamanaka genes, 3 of them, for a long time, really, without having any problems with the toxic effects or tumors, teratoma, which is something that typically happens in transgenic mice for the Yamanaka. Well, even then, constructed a mouse, a transgenic mouse, expressing the 3 Yamanaka genes, they used OSK. So when they gave doxycycline to the drinking water of these animals, there were very, very toxic effects. And in 4 days, more or less, the animals died. So that was really a paradoxical finding, because when administered, when the delivery was made using a viral vector, in the neurons at least, in the nerve, the effects of the long-term expression of the transgenes, the Yamanaka transgene, was quite regenerative, quite benign, without any kind of pathology. But when they express the same genes in a transgenic animal endogenously, the genes were really very, very toxic. And this is, in fact, in line, this kind of dual behavior of the Yamanaka genes according to the way they are delivered to animals in vivo. And this is in line with an ongoing experiments we have in our laboratory. We're working with primary cultures of human umbilical cord cells. This is a typical culture.
Rodolfo,
yes?
just sorry to interrupt you, but be careful because normally in 3 minutes you should finish, and then we have already no time for questions. Yeah.
Okay. So when we expose the cells to an adenoviral vector expressing the 4 Yamanaka genes, and for 7 days, we found a marking distribution of the cells, and most of the cells are expressing the transgene. This is why they are fluorescent. And even 85 days after infection, they're still alive, and they are expressing the transgene. So the main point here is to show that the delivery of the Yamanaka genes to these cells can be also kept for a long time without any adverse effect. Well, I am very short of time, so I will very briefly stop here when I need to stop. We are doing an experiment, a life extension experiment on female rats that are 25 months old at the beginning. We're using 2 groups, control groups that receive nothing, and an experimental group that receive young plasma every other week. And these are represented by the solid symbols. And they are, this is a survival curve. We are 11 weeks into the treatment. Before the study, we perform a memory test. And on week 12, we will perform another memory test to compare the response of the 2 groups. We are also taking blood samples every other week to measure epigenetic age. So I will this is the last description I will make. This is a pilot experiment we did several years ago. In that experiment, we used a single rat, female rat, 6-month-old adult rat, and we treated the rat with human mesenchymal stem cells, intravenously given every other week. And the idea was just to have the pilot experiment running for 4 weeks and then do a more complete experiment. But things work in a different way that we expected. At this pilot experiment ended up being published, and this is the final slide, I think. These are the survival curves of male rats in our colony. These are the survival curves of many rats, 100 rats. Of female, they live longer. This is the maximum lifespan recorded in the literature for albino rats, 36 months. And our treated rat lived up to 44 months. So 8 months beyond the maximum lifespan. This is quite a remarkable achievement that we didn't expect really. So this is why we decided to publish that result. And this is the last image. This is the long-lived rat, 1 month before death, a control rat, old rat, and a young one. Sorry for taking so much time. Thank you very much.
Thank you, Rodolfo. I will read the questions of Magomet Kaidakov. My concern with epigenetic rejuvenation is its potential effect on previously accumulated memories. It has not been tested yet. If life experiences individuality are compromised as a result of epigenetic rejuvenation, what then? What do you think about this question?
Can you repeat that because it wasn't?
It's in the chat also.
Oh, okay.
Okay, if you can read it.
Yeah,
while let's say while you read it, I will already ask to Harold to share his screen, and maybe Rodolfo, you can stop your screen, or
I am trying to do this.
Or somebody can do it also. I know I can do it.
If you can do that.
I don't know where. If just a second, technical. Yes.
Okay.
Okay. Yeah.
Okay. So I'm looking for the chat. Sorry, I am not familiar with.
Okay. I will read it again. My concern with
question. Sorry, guys. So we are behind the time, or what's the schedule?
What?
And what is the protocol? Can I ask a question, or add all questions in chat and then Didier?
No. Normally you can ask. So ask your question. It will be easier, and then we give the floor to Rodolfo. Yeah, go ahead.
Cool. So so my question is, the control rats, how were they controlled? Were they injected with something else? Sorry if I missed that point. And then the second question is, you know, sorry, I will just wait until you answer this part. How the control rats treated?
The plasma, the rats treated with young plasma, Irina, you mean? Those control rats?
What the control rats receive?
The control rats in the plasma life extension experiment are not receiving anything. Just they are
right. So then my hypothesis would be that the rats which are receiving something and are being treated, they are really not identical to the rats which are not being treated. They're being picked up, they're undergoing this experimental procedure. And there are numerous neuropeptides, for example, oxytocin, on which we published, which could promote rejuvenation.
Yeah, right.
So they are basically, you know, it's kind of the proverbial placebo effect in people, but it is not just placebo. It's actually different signaling in your brain and different release of the peptides, which improve metabolism and, you know, tissue health and repair. So if the control rats were treated with some other, you know, positive fraction lacking plasma, or I don't know what else would be a good treatment, because if you simply do saline plus albumin, you are actually treating them by diluting their agent secretome. Something like that, sorry.
Yeah, we are having another group of old rats, control rats, receiving PBS, but these belong to a separate experiment, but they are part of the same group of rats. And we are having doing the survival curves for all rats for us, for our internal use. And those rats receiving PBS for several weeks now, the survival curve is exactly the same. So what I didn't mention.
Not good for them.
Sorry,
PBS
Irina.
is not good for them, right? It will make them less healthy. Anyways, just something to think about.
Sorry, Irina, sorry, Rodolfo, but we are already ahead of time.